In a biology lab on the campus of Eastern Illinois University, an important component of COVID-19 specimen collection is being prepared. It's called viral transport media, or VTM.
It's very similar to the stuff I use in my cell culture to grow cells, but this isn't a growth media, it's more of a stabilization media, so that if you put a biological sample into it, it should not degrade, at least not too quickly. And so that's the idea of viral transport. You're not trying to grow or to kill the virus, you're trying to stabilize it until you can get it to the lab so that they can do an analysis on it to see if it's, in this case, for COVID-19, if it has the virus or if it does not.
VTM is in short supply because it is only produced in Germany and Italy. When the Centers for Disease Control published the VTM formula, Sarah Bush Lincoln reached out to EIU to see if they might be able to help.
Yeah, we had the great idea to contact Dr. Bulla at Eastern, and he told us that he would contact some of his colleagues there, and our lab operations manager, Dave Sowers, coordinated that phone call with some of the people at Eastern, and Dr. Bulla got their department involved in looking at what capabilities they would have. So they used the recipe and determined they would be able to make about 3,000 of the viral transport medias for us.
Yeah, we've had several individuals from the Department of Chemistry, Dr. Beck, Perianen, Treadwell, and Chestnut, folks from Biological Sciences, Dr. Kang, Dr. Hung, myself, and then we also are bringing in some graduate students into the project.
Once everything was in place, the team at EIU created a 10-liter batch of VTM.
It's just a matter of gathering the reagents, combining them together in the right proportions, making sure everything is going through sterilization techniques.
With the VTM ready, the real work begins. 3 milliliters is dispensed into vials, one at a time, under sterile conditions. When complete, the team at EIU will fill 3,400 vials.
It's a fairly basic process, but you just have to be very cognizant that no contamination enters along the way. We're very glad that they allowed us to take part in this. Right now we're doing, of course, teaching and research and all that stuff like we're normally doing, but it's very good to get onto this big COVID-19 thing because it's such a worldwide problem. It's very good that we're able to contribute to that in some way.